For decades, carbohydrates have often been portrayed as a primary dietary villain, blamed for metabolic dysfunction and systemic inflammation. Yet, across the globe, populations living in regions known for longevity consume carbohydrate-rich diets as staples. How can this apparent contradiction exist? The answer lies in a specialized class of carbohydrates that behaves unlike typical sugars. Known to scientists as resistant starch, this unique molecule bypasses early digestion entirely. Instead of causing rapid spikes in blood glucose, it travels intact to the lower gastrointestinal tract. Found naturally in foods like green bananas, legumes, and cooked then cooled starches like potatoes and rice, resistant starch is reshaping how researchers view dietary energy. Could this overlooked compound hold key insights for metabolic health, immune modulation, and healthy aging? Let us explore what decades of nutritional science actually reveal about the mechanics of resistant starch. To understand how resistant starch functions, we must examine its distinct cellular interactions. Unlike standard amylose and amylopectin, resistant starch possesses a molecular structure that resists enzymatic cleavage by alpha amylase in the upper gastrointestinal tract. Passing into the large intestine, it becomes a specialized substrate for resident colonic microbiota. Through bacterial fermentation, microorganisms convert this starch into short-chain fatty acids, primarily acetate, propionate, and butyrate. Butyrate, in particular, serves as the preferred energy source for colonocytes, the epithelial cells lining the large intestine. Research suggests that these short-chain fatty acids support cellular junction integrity, strengthening the gut mucosal barrier. Furthermore, these metabolites interact with specific cell membrane receptors, such as free fatty acid receptor 2, which may influence signaling pathways involved in systemic inflammation, insulin sensitivity, and postprandial glycemic control. This microbial conversion transforms a complex carbohydrate into active metabolic signals throughout the human body. The scientific literature regarding resistant starch spans several tiers of evidence, requiring careful distinction. In vitro laboratory cell assays demonstrate that butyrate directly stimulates cell proliferation and reinforces mucosal protein structures in cultured human intestinal cells. Moving to animal models, rodent studies show that diets supplemented with resistant starch reduce markers of systemic oxidative stress and improve lipid profile markers. However, findings in animal models do not automatically translate to human outcomes. In human observational cohort studies, populations consuming high-fiber diets containing resistant starches consistently show lower incidences of metabolic disorders over multi-decade tracking periods. Crucially, randomized controlled human clinical trials provide stronger insights. In various 6 to 12-week double-blind human trials, participants consuming 10 to 30 grams of resistant starch daily exhibited modest improvements in insulin sensitivity and reduced post-meal glucose spikes, though individual responses vary significantly based on baseline gut microbiome diversity. Translating this scientific research into practical nutrition requires understanding food structure and preparation techniques. Naturally occurring sources include slightly green bananas, lentils, and beans. Interestingly, cooking and subsequently cooling starchy foods like potatoes, oats, or rice creates a chemical process called starch retrogradation. As cooked starch cools, its molecular structure recrystallizes into a form highly resistant to digestive enzymes. In human studies, dietary interventions have examined intake levels ranging from 10 to 30 grams of resistant starch per day, usually introduced gradually. Increasing resistant starch intake too rapidly can lead to transient digestive symptoms, such as abdominal bloating or excess gas, as gut microflora adapt. While resistant starch is generally safe for most people, individuals with severe digestive conditions or irritable bowel syndrome should introduce these foods cautiously. Furthermore, people taking medications that affect blood glucose levels or gastrointestinal motility should discuss significant dietary changes with their healthcare professional. In summary, current scientific evidence indicates that resistant starch is far more than a simple source of calories. By resisting early digestion and nourishing beneficial gut bacteria, it produces short-chain fatty acids that support gut mucosal integrity, metabolic health, and balanced inflammatory pathways. While resistant starch is not a standalone miracle solution or direct cure for age-related decline, incorporating sources like legumes, green bananas, and cooled starches can be a valuable component of a balanced, nutrient-dense diet. Science reinforces that individual responses depend heavily on baseline health, lifestyle factors, and unique microbiome compositions. This video is for educational and informational purposes only. It does not constitute medical advice, diagnosis, or treatment. Always consult a qualified healthcare professional before making significant changes to your diet, lifestyle, or medications.